Good afternoon, everyone. My name is Kendall Smith, and I'm with the Department of Anesthesiology at WashU and St. Louis. And I'm really excited to kick off today's session, talking a little bit about molecular circadian rhythms and postoperative delirium. I have no disclosures. So postoperative delirium is characterized by an acute and fluctuating course of a constellation of disturbances in neurocognition. And unfortunately, it commonly affects older adults undergoing major surgery and anesthesia with increased morbidity and mortality. Its pathophysiology is poorly understood, but there likely is a more multifactorial etiology. And today I'm going to focus on circadian rhythm disruption as a putative contributor. Circadian rhythms are vital to almost all life on Earth, and we have internal rhythms that receive external cues that help us adapt to our environment. And light is one of those external cues that provides information through the retina to the suprachiasmatic nucleus, which is thought to be the master clock of the body. Information is then relayed down the CNS to peripheral organs, each of which have their own clocks to help manage and coordinate internal processes. Now, both in the CNS and in the periphery, a core set of clock genes governs this process and regulates transcription through a series of positive and negative feedback loops. And Doctors Hall, Rosbash, and Young are responsible for elucidating the machinery of the molecular clock, and for that, they received the Nobel Prize in 2017. Now, it turns out it's not just the clock genes that oscillate, but that 30% of the human transcriptome actually oscillates in circadian fashion. And we think that this is how potentially organ systems are able to coordinate with each other to optimize physiology and function. One of the things that I'm really interested in doing is understanding the impact of major surgery and anesthesia on circadian rhythms, and then also understanding relationships to perioperative brain health. Circadian disruptions have been linked to both acute and chronic CNS disorders, including depression, Alzheimer's disease, and related dementias. And we have seen associations between abnormalities and circadian rhythms in terms of their amplitude, increased fragmentation, as well as extremes in phase angle when patients are either early or late chronotypes or are morning larks or night owls. Actigraphy is commonly employed for examining these rhythms, and this is a study of rest and activity using accelerometry. And another assay that's commonly employed is dim light melatonin onset, which measures melatonin in the evening under specific dim light conditions, and that helps us understand chronotype. We also know that general anesthesia is a clock disruptor, and significant delays have been observed after general anesthetics in honeybees and mice. And there's a very interesting clinical trial ongoing in humans in the Warman lab at the University of Auckland. Studies linking acute circadian disruption to postoperative delirium have also been performed, but thus far they've been relatively smaller in scale. And some of them lack some granularity in the sense that they use melatonin or actigraphy as indirect measures of circadian rhythms. You can imagine that in a hospital setting, this is actually sort of challenging to implement. Our patients aren't as mobile as normal, and also significant artificial light in the hospital setting makes it difficult to employ and implement an assay such as dim light melatonin onset. So what I would like us to do is to think about rhythmic gene expression as a both more feasible and comprehensive measure of circadian integrity. Time signature is one algorithm that proposes to do that. So in a nutshell, you will provide time signature with gene expression data from two different time points, and time signature will spit back out an internal predicted time. Now, graphically speaking, this is sort of what that might look like. So we're looking at a control cohort of young, healthy patients under control conditions. And on the x-axis, we're looking at true time. And you can see that these patients were, their blood was sampled quite frequently every 2 hours. And on the y-axis, we're looking at time signature derived transcriptomic time. And I hope you can appreciate the time signature is fairly accurate. So all the dots that are landing within the dark gray bands denote accuracy within 2 hours. And then those landing within the light gray bands denote accuracy within 4 hours. And there are a number of advantages to time signature, but one is that it's generalizable and that it can be applied to across gene expression platforms without the need of extensive recalibration. And the second is that it requires as few as 2 blood samples from a patient suitably spaced. And so the thing that I'm really excited about is how we can leverage this type of algorithm in the perioperative and inpatient settings. But the first thing we need to do is determine whether or not time signature would even work in older adults. And so in a collaboration with Drs. Braun, who is the developer of time signature from Northwestern University, along with Dr. Ju and Dr. Music from the Department of Neurology at WashU, we recruited 40 community dwelling subjects who were 65 years or older. We performed RNA sequencing and time signature application on blood that was sampled in the morning and the evening. We also collected wrist actigraphy data from these patients and also brought them in and performed a dim light melatonin onset on them in our sleep lab. We also had them complete a Horn-Ostberg morningness eveningness questionnaire, which allows us to give a subjective measure of chronotype. And we were very pleased to find that without the need for retraining, time signature did maintain accuracy in older adults to a level comparable to what had been previously published. So again, looking at true time here on the x-axis, where we sampled blood around 10 in the morning and then also around 8:30 at night, and compared that with transcriptomic time derived by time signature here. You can see that most of the dots do land within the dark gray dots, and we got dark gray bands, and we did get an acceptable normalized area under the curve. So we also computed a variable, which is to say we were wondering what was the significance of the difference between the true time and the transcriptomic time. So we coined that variable transcriptomic angle. And so we first compared the morning and the evening angles, and we did find that there was a significant correlation demonstrating algorithm robustness. And then we began to hypothesize that perhaps that data spread that we're observing here might represent chronotype. And so you can imagine that individuals whose internal clocks are later than local clocks, maybe those are morning larks. And individuals whose internal clocks are delayed relative to true time might be night owls. And in fact, that's what our data really kind of suggested when we related the transcriptomic angle to traditional circadian measures. So we related it to actigraphy, DILMO, and MEQ score. And what we found is when we look at the actigraphy data that individuals who were predicted to be morning larks, according to the transcriptomic angles, they're also waking up earlier, they were starting their day earlier, and they were going to bed earlier. Similarly, according to the DILMO assay, they were secreting melatonin at an earlier time, and they also were self-reporting to be morning larks, according to the MEQ score. So we were really excited about these data because, what I think that this might suggest is that time signature might not only be able to determine phase, but might also allow us to phenotype individuals in relation to their phase. So at this point, I'd like to transition our talk into the perioperative realm. And I want to talk about a really exciting study that I am starting with the support of the IARS Mentored Research Award. And what I really want to do is now that we understand that time signature really might work and might be ideal in the perioperative setting, I want to look at transcriptomic circadian rhythms and then also relate it to postoperative delirium. So the objective of the study is to characterize transcriptomic rhythms in older adults undergoing elective cardiac surgery and then evaluate relationships to postoperative delirium severity. So this study builds off of an R01 funded study in the laboratory of Ben Falanca called PDRowsy. And PDRowsy is focused on understanding EEG markers of sleep and wakefulness in relation to delirium and delirium severity. And what I really want to do is add this molecular arm to that study. And so what we're doing essentially is we're taking longitudinal measures of EEG and delirium assessments as well as blood draws before cardiac surgery and serially after cardiac surgery. And I wanted to share with you a couple of schematics of what a hypothetical patient might look like in terms of transcriptomic rhythms. And so let's look at hypothetical patient A. And we hypothesize that maybe we would find a phenotype or a cohort of patients whose rhythms might look like this patient, where perhaps preoperatively the rhythms are closely aligned with true time, which is also sort of denoted by the reference blue waveform. And postoperatively, perhaps we do see that there is a slight change in transcriptomic angle, but perhaps it's not terribly significant. Now compare that with hypothetical patient B, whose preoperative rhythms actually might denote some type of angle before surgery, but postoperatively, perhaps we'll identify patients who have significant phase shift or change in that transcriptomic angle. We hypothesized that perhaps patients who have a smaller magnitude of change might have better neurocognitive outcomes, and patients with a more significant magnitude of phase shift might have worse perioperative and neurocognitive outcomes. And so I'm only 4 months into this award, and we're still working feverishly to collect data, but I did want to show just some preliminary data that I found interesting to maybe sort of make some observations from. So far we have been able to longitudinally sequence 12 individuals before and after cardiac surgery in the morning and the evening. And these are the data on the day before surgery. And so I'll just draw your attention to the normalized area of the curve, which is 0.77, which is not too bad. And as you can see, kind of most of these dots are landing within either the dark or the light bands. Now compare that with the preliminary data that we have from postoperative day 1. And just kind of to the naked eye, you can tell that these transcriptomic rhythms do appear to be significantly disrupted on postoperative day 1. Now, there are a number of ways that I think we could sort of preliminarily interpret these data, but at the moment, I'll just sort of focus your attention on these PM time points because this PM preoperative time point did appear to be a little bit cleaner than the AM. And so if we look at those data in a slightly different way, I think we can sort of make some interesting preliminary observations. Here on the x-axis, we're now looking at each individual that we sequence. And on the y-axis, we're looking at the time signature derived transcriptomic angle. And I think it might be helpful for me just to walk you through one of these individuals to sort of help you make sense of these data a little bit. So this is individual 2, and the green dot up here denotes the preoperative transcriptomic angle, which is estimating that the patient's preoperative internal clock is about 4 hours ahead of local time. Compared that to the postoperative angle in the same individual, which appears to be about 1 hour behind local time. And so what that says is that this patient in particular had about a 5-hour phase shift between the day before surgery and the day after surgery. If we look at these data a little bit further, we can make some other interesting observations. 9 out of the 12 individuals actually did exhibit a phase delay. And 3 of those, the phase delay was actually quite pronounced. And so if we think back to our schematic, perhaps we are beginning to see several cohorts or phenotypes of patients emerge, where some patients have maybe a relatively smaller magnitude of change, and other patients have a significant phase shift. Now, whether or not this is related to delirium severity is going to be in my future plans, and I'm really excited. That's part of my aim too. So please stay tuned for that. So we're also interested in continuing to optimize the time signature algorithm or continue recruitment and data collection. We're also really excited about looking at later time points because in addition to looking at postoperative delirium severity, we're interested in perhaps looking at transcriptomic signatures as potentially a marker of delirium resolution. We also want to look at relationships to longer-term neurocognitive outcomes and also how to integrate this rich RNA-seq data set with EEG markers and other markers of inflammation and neurological disease. So in summary, I hope that I've been able to convey to you how important circadian clocks are in helping us coordinate complex internal processes in the brain and the periphery, and how transcriptomic circadian phenotyping in particular in older adults might be a way for us to estimate circadian function perioperatively. And my preliminary work shows that molecular rhythms are acutely disrupted on the day after cardiac surgery. In our future work, we're really excited to look at relevance to postoperative delirium and other perioperative neurocognitive disorders. And with that, I'm really happy to take any questions during the discussion section. I want to thank this amazing team who has helped me facilitate this project, including the Department of Anesthesiology, my boss, Dr. Michael Avedon, who's a tremendous mentor and who inspired me to do this project, Drs. Ben Polanka, Dr. Pratik Sinha, Drs. Music and Yu from the Department of Neurology, as well as Dr. Rosemary Braun from Northwestern. I want to thank all of the funding mechanisms that has helped to make this happen, and in particular the IARS, who awarded me the Mentored Research Award last year. So thank you very much.
Hi, everyone. Thank you for having me, and thank you, Miles, for organizing the session. It's a pleasure to be with you all today, albeit virtually. I hope you're all staying safe and healthy. So today I'm going to be discussing caffeine and postoperative delirium. And broadly speaking, brain health in surgical patients really is a hot topic right now. The ASA has launched the Perioperative Brain Health Initiative a few years ago, and the American College of Surgeons have also launched the Geriatric Verification Program. And so as a society, I think we've realized there's a programmatic need to optimize care for older, vulnerable patients entering the hospital. And this is where advancing our neurobiological understanding of delirium and developing neurobiologically informed therapies, I think, are incredibly important here. So of note, our research team in our institution receives support from Blue Cross Blue Shield for delirium quality improvement initiatives that are otherwise unrelated to the work I'm presenting today. And we did receive an IND exemption from the FDA for testing the effects of caffeine for improving postoperative recovery, but otherwise no conflicts of interest to disclose. So I want to start first by framing the context of the problem our group has been trying to address. So many of our surgical patients enter this refractory perioperative cycle of adverse pain arousal state connections that's hard to break. And so often our patients come in having reported not sleeping well the night before, right? I mean, they might be driving in from far away. They might have been anxious or nervous, but we commonly hear patients don't sleep well the night before surgery. So we start there, and then we know that a lot of patients still experience moderate to severe pain after surgery, and opioids are still a mainstay therapy for pain postoperatively, and both pain and opioids can then disrupt sleep architecture. So we have pain disturbances that can also, sleep disturbances that can make pain worse. And then with sleep disturbances, pain, and opioids, all of these are risk factors for delirium, and all of these make cognitive function worse. So a fundamental question then becomes, how do we break this cycle? Are there any biologically informed therapies that we have that might plausibly work? And one possibility is a drug that many of you might be using right now, and that is caffeine. And so from an analgesic perspective, there are basic science data from Giancarlo Vanini and his team here at Michigan demonstrating that caffeine might confer both acute anti-nociceptive properties and also reduced hyperalgesia for up to a week after surgery in a rodent model. And there's also a Cochrane review highlighting a track record of analgesic efficacy with caffeine when it's used as an adjuvant after outpatient surgery. And we also know that caffeine enhances arousal and improves various domains of cognitive function. So it's conceivable that caffeine could ameliorate this perioperative cycle in nuanced ways, particularly given that both pain and sleep deprivation are risk factors for delirium, like I mentioned. So that's really the background and framework behind why we conducted this trial, where we tested the central hypothesis that caffeine would improve postoperative pain and related neuropsychological outcomes, including postoperative delirium. And so this was a small 60-patient single-center trial we conducted at Michigan Medicine, where 30 patients were randomized to placebo, and then 30 patients were randomized to 200 milligrams of IV caffeine citrate, and the study drug was given during surgical closure. And this was for adult surgical patients presenting for laparoscopic GI and colorectal surgery. And the dose of 200 milligrams was really informed by the pain literature, since this is a pretty common dose for treatment of posterior puncture headaches. It's also a dose that was in the range studied with that Cochrane review that I mentioned a moment ago. So the primary outcome for the trial was actually cumulative postoperative opioid consumption for the first 3 postoperative days. And as you're seeing, based on our pre-specified GE modeling, the estimated difference between groups was actually 55 milligrams more median oral morphine equivalents in the caffeine group compared to the placebo group. And so this didn't quite reach statistical significance, but there was perhaps a trend that way. So by chance, there were also some baseline group imbalances. So for example, and pertinently, the placebo group, there were more open conversions from the laparoscopic approach, which of course can increase pain and opioid consumption. So after we adjusted for confounders like that, we actually did find that there was an adjusted difference. There was an adjusted increase of median oral morphine equivalents in the caffeine group spread out over the first 3 postoperative days, which is of course discrepant with our original hypothesis. For what it's worth, there were actually no differences in subjective pain scores throughout each of these time points. And the increased opioid consumption really seemed to be confined to the PAC use setting. That's when really we saw the increased opioid consumption. So, why is that? Is it that caffeine caused increased pain and drove increased opioid consumption? It's possible. What I think is more likely was that participants who were randomized to caffeine were more awake and alert and able to effectively communicate pain to the PACU nerves. And in fact, we saw that for participants randomized to caffeine, there was a reduced risk of PACU delirium based on our confusion assessment method screening 1 hour after PACU arrival. So for the caffeine group, the proportion of patients who screened positive for delirium was 12% versus 33% in the placebo group. And this was concurrent with increased frontal parietal connectivity based on whole scalp EEG analysis we were doing at the time. Now, we had pre-specified postoperative delirium as a secondary outcome, but we had specified that through the first 3 postoperative days, including the PACU. That wasn't significant, but was it was right on the threshold of significance, which caused us to look at the delirium prevalence daily, and that's where we noticed this post hoc reduction of PACU delirium prevalence. But it did occur, as I said, with these EEG changes. And this is important because we know that frontal parietal connectivity is important for cognition and consciousness. So they're intriguing findings. The problem here is that EEG connectivity measures are nice, but they don't tell the full story. And in fact, we've seen situations of increased frontal parietal connectivity during general anesthesia, for example. And so just to give you an example, this was a separate observational study from our team conducted a few years ago to identify connectivity states during general anesthesia. And that we found during the anesthetic maintenance phase, there were actually periods of relatively high alpha frontal parietal connectivity during general anesthesia. And these were values that were very close to the baseline preoperative values here. And we also know that during certain pathological states, like seizures, for example, we can see pathologic states of hypersynchrony and increased connectivity. So again, focusing just on connectivity really doesn't tell the full story. So in this context, we wanted to test the concept of neural criticality in patients experiencing PACU delirium versus those without delirium. So what is criticality, and why specifically are we focused on this? So criticality is the state of a system when it is poised to dynamically shift based on external stimuli or input. And the classic example to illustrate this is this sand pile thought experiment, where someone is dropping grains of sand slowly on a pile, and eventually the pile becomes so unstable that the next grain of sand causes it to collapse in an avalanche. And this pile system is sort of naturally organized this way. It doesn't require any external tuning parameters, so to speak. And indeed, natural systems actually do seem to fit this model. We see that real avalanches do actually fit this model of criticality with accumulating snowfall. What's interesting is that this criticality property could actually be advantageous for biological systems as well, like the brain. Because if the brain is tuned or poised at that critical state where it's about to shift, this might confer the benefit of being able to flexibly shift among different brain states for meeting demands. And in fact, there are fMRI signatures of criticality that correlate with cognition in human volunteers, and impaired criticality actually correlates with both Alzheimer's disease presence and progression, which is worth noting. And again, these are based on fMRI signatures of surrogate measures of criticality. So we performed a secondary analysis of our clinical trial where we sought to determine whether EEG-based biomarkers of criticality might be associated with pachydelirium. And so there are two EEG-based surrogate biomarkers, or at least candidate surrogate biomarkers that we tested. And so the first is autocorrelation function at the top here. And this is where we are determining if a neural time series serially correlates with itself over time. And a higher autocorrelation function, and that's kind of what we see here in orange, a higher autocorrelation function implies proximity to criticality, because this serial correlation suggests intermittent synchrony and loss of synchrony over time, presumably as networks are flexibly engaging and disengaging as needed based on external input and task requirements. So the second biomarker here is the topology of phase lag entropy. And phase lag entropy reflects the diversity of signal synchrony throughout the brain. And in this example here at the bottom, so these are average data from a group of awake, healthy human volunteer research participants. And what you're seeing here in the posterior part of the brain, and this is a whole scalp EEG system, the blue is essentially showing us low phase lag entropy power, low phase lag entropy. And this is meaning that we're seeing low diversity in phase synchrony, which means that there is actually a high degree of phase synchrony here. And we know that these posterior brain regions are really important for consciousness. They have a lot of important network hubs with rich connections that are important for consciousness. And so the implication here is that there's going to be high synchrony and a relatively low signal diversity that might be important for this region. What's important to point out is this sort of anterior to posterior asymmetry of phase lag entropy correlated strongly with criticality based on computer modeling studies led by Dr. Anshul Lee and his group here at Michigan. So we wanted to test these surrogate biomarkers in our clinical trial patients. So we applied these techniques for patients who were randomized to placebo and caffeine, and here's essentially what we found. So on the left in this spectrogram, these are patients who were ultimately delirious in the PACU. And what we found, and this is circled here in red, is that this blue shading represents significantly reduced alpha autocorrelation function in patients who screen delirious based on the confusion assessment method. And we know that alpha rhythms are important for consciousness. And in fact, alpha autocorrelation function specifically helps to coordinate long-range coordination of neural processes for sensory integration and cognition. So alpha autocorrelation function was significantly reduced during delirium. And in fact, we saw a shift towards theta autocorrelation function. So there was a shift towards slower wave rhythms and slower wave criticality. And then over here, we see this coherent red signature here in patients who were randomized to caffeine. So caffeine, we knew it reduced pachydelirium. It also seemed to increase alpha autocorrelation function. And this y-axis here, this is the PACU time. So in terms of phase lag entropy with delirious patients here on the left, we sort of see this loss of that anterior to posterior asymmetry pattern, but in patients who weren't delirious, we see some preservation of that pattern here, where there was a bit higher entropy anteriorly, and then again in the posterior regions, phase lag entropy was lower, with the inference being that synchrony was higher in these posterior regions. Likewise, in the placebo group, we see low phase lag entropy kind of globally throughout the brain, even centrally. But with caffeine, we see this gradient is sort of restored, lower phase lag entropy, potentially higher synchrony in the posterior nodes, and then higher diversity of signals in the anterior brain regions. Scales are a little bit different, but it does seem that this asymmetry was better preserved in the caffeine patients. So, this, just to highlight, this was a secondary analysis of a small trial. We're still testing these biomarkers. So I think this is still hypothesis generating. But I do think the concept of impaired criticality with neurocognitive disorders is plausible, based on our empirical findings and modeling studies to date. So I'll wrap up there, but I do just want to highlight delirium is a major public health issue. The pathophysiology is still poorly understood, and that's why I think it's for us as a clinical and scientific community to help further advance our pathophysiologic understanding and develop advanced neurobiologically informed therapies. So I'll wrap up there. I would love to thank our Division of Perioperative Neuroscience, who were immensely helpful with getting us these data. Happy to take any questions. Thanks so much.
Hi, it's a pleasure to be with you here today, even virtually. I'm Miles Berger, and I'm going to talk to you today about the role of the default mode network in perioperative neurocognitive disorders and delirium. Here are my disclosures. Basically, no financial support from industry, no off-label or investigational drug use. So here's what we're going to talk about today. First, we're going to go over what delirium and perioperative neurocognitive disorders are. We'll do a little bit of background and make sure we're all on the same page. Then we'll talk about what the default mode network is. And third, we'll talk about what the role of the default mode network is in delirium and perioperative neurocognitive disorders. So here's a paper from Sharon Inouye's group from about 6 years ago. And I'm going to point out a couple things about this graph in this paper that help illustrate, I think, why it's reasonable to think of delirium and cognitive dysfunction following surgery as sort of a spectrum or as two different sides of the same coin. So what Sharon and colleagues did here is they did cognitive testing on a bunch of individuals before and after surgery at various time points, and they screened them for delirium after surgery. And they grouped the patient's cognition separately by those who developed delirium in blue versus those who didn't develop delirium in red. And what you can see here is that the patients with delirium in blue show an early or acute drop in cognitive function on a global cognitive performance scale. But then they recover, have this mid-course correction. But then longer term, by 3 years later, they show worse cognition. And that's actually quite similar to what Mark Newman and colleagues at Duke showed 15 years earlier. They did cognitive testing, not delirium screening, but just cognitive testing on a large cohort of several hundred cardiac surgery patients. And what they found was actually very similar to what Sharon and colleagues found 15 years later. There's an initial drop in cognition that some patients have, whether you identify them by delirium or whether you just identify them by decreased performance on cognitive tests. But then both groups show an improvement back to their baseline in the sort of interim or intermediate recovery phase. But then longer term, 3 to 5 years, patients who had a cognitive problem early after surgery, whether it was defined by a delirium assessment tool or simply by cognitive tests, those patients tend to have worse cognitive decline 5 years later, worse cognitive decline 3 years later. So this similar trajectory between the two disorders is part of why we think of them as basically two different faces of the same coin. And that's what led Jeff Silverstein and colleagues some years ago to create this figure from a review article they published in Anesthesiology that emphasizes that these disorders are really a spectrum. We typically see postoperative delirium a couple days after surgery. Post-op cognitive dysfunction is typically measured now. We think of it as 1 to 12 months after surgery. But either way, there's no acute dividing line where a patient suddenly goes from one into the other. It's more of a spectrum, at least when one thinks about the mechanisms that are likely involved in these disorders. Okay, so now let's talk about the default mode network. And then once we do that, we can talk about its role in these disorders. So this is a picture from Marcus Rakely and colleagues from several review articles they've published. And these yellow and orange hotspots are key hubs of the default mode network, the posterior cingulate, the anterior cingulate, as well as some of the areas of the medial temporal lobe. And the default mode network is basically a set of brain regions that show correlated activity patterns and that show activity when a subject is at rest when they're not completing a cognitive task. This includes the posterior cingulate and precuneus right around here, the anterior cingulate and superior frontal gyri, as well as the angular gyri that you can see nicely here in cross section, and the superior frontal gyri. So it's a set of brain regions that are both midline and bilaterally symmetric regions that all show this correlated activity when somebody's at rest. Now, okay, I've told you what it is, but what does the default mode network do? Well, we don't have a perfect answer to that yet. What we can say is that the DMN, these correlated activity patterns between these specific regions, this exists across mammalian species. It's not unique to humans. It's what we call a task negative network, meaning that these brain areas are active, and they're active in correlation with each other when someone is awake but resting and not really doing anything, when they're not completing a cognitive task. In that sense, since it's going on or it's active when one is not doing anything, that's what led to the title for this talk, A Brain Network About Nothing. Just like Seinfeld was a show about nothing, the DMN is a brain network that's involved when you're not doing anything. Now, DMN activity is suppressed by volatile anesthetics, at least at GA doses. Ben Polanka and colleagues showed that about 7 years ago now. And DMN activity and connectivity is reduced in mild cognitive impairment and in Alzheimer's disease. A number of people have shown this multiple times. Interestingly enough, amyloid pathology, one of the core pathologic hallmarks of Alzheimer's disease, begins in the posterior cingulate and spreads initially within the default mode network. So we don't completely understand what it does, but hopefully this gives you some sense for what scientists in the field are thinking about when they think about what the DMN does. So now about 5 years ago, there was a group in Australia, Gideon Kaplan's group, that did PET scans in patients who had delirium and then after their delirium resolved. And what you can see here is in the delirious brain, there's profound hypoactivity, hypoactivation in both the anterior cingulate and in these posterior cingulate areas, which improved to a great extent after delirium. And when they quantified this, they found that there was a decrease in brain activity via PET scans across the whole brain, a small percentage decrease, but there was an even larger decrease in brain activity during delirium in the posterior cingulate bilaterally, and that improved after the delirium episode resolved. And those effect sizes were roughly twice the size of the decrease in activity across the whole brain. So this tells us that the delirious brain is less active, and in particular, a key hub of the DMN, the posterior cingulate, is particularly less active during an episode of delirium. Now, even earlier, Choi and colleagues had published this work using functional MRI. And functional MRI is a technique that basically, it's like MRI that we all know about and see MRI scans clinically, but it parcellates the brain into these 3 cubic millimeter voxels or cubes. And what they did in this paper is they asked if we place a seed electronically in one particular brain region here in the posterior cingulate, and we asked the computer to tell us which other brain regions show correlated activity patterns with the posterior cingulate. Well, I've already told you it's part of the DMN, so you would expect what we see here in the controls or comparison subjects. You see a lot of connectivity between the posterior cingulate, the precuneus, and the anterior cingulate, as well as these angular gyri, et cetera. This is the default mode network. And it's anti-correlated with visual cortex function here and with some of these dorsolateral frontal regions. However, in the patients with delirium, you can see that the posterior cingulate is much less well connected to the precuneus and even more greatly decreased in connection to the anterior cingulate. And you can see that nicely here. And then what they went on to show in this paper, very interesting paper, is that the connectivity strength between the precuneus and the posterior cingulate, 2 key hubs of the default mode network, is related to both delirium severity and duration. And so what they've done here is graphed in blue and red, that's just the left and right sides of the brain, the connectivity strength between the posterior cingulate and the precuneus, that's on the y-axis. And on the x-axis, they've got delirium severity. And you can see there's an inverse correlation. In other words, the more connected these 2 regions of the DMN are to each other, the less severe the delirium is. And conversely, the less connected these 2 brain regions of the DMN are, the greater the delirium severity is. And they see the same thing when they look at delirium duration. Again, greater connectivity within the DMN, within these 2 brain regions, is associated with shorter delirium, and lesser connectivity strength between these 2 DMN regions is associated with longer delirium duration. And then our group has looked at this as well in patients after surgery displaying cognitive dysfunction after cardiac surgery. And what we've done here is in an fMRI analysis, we looked at the activation strength and connectivity of different regions in the brain in patients who either develop cognitive dysfunction after surgery and in similar folks who didn't have surgery and didn't have cognitive dysfunction. What you see here is quite interesting. In the blue triangles, we have the cardiac surgery patients. And what you can see is the greater their drop in connectivity strength to the posterior cingulate, the greater their drop in cognition is. Conversely, if they didn't have much of a drop in connectivity within the posterior cingulate, they didn't tend to have much of a change in cognition. Vice versa, in the non-surgical controls, there's no real relationship. And partially that's because there's just not a lot of cognitive change in the non-surgical controls here, nor is there much change in connectivity in this brain region. So again, it points to, I think, a decrease in connectivity within the posterior cingulate, this key hub of the DMN, in patients with cognitive dysfunction following surgery, as well as in those with delirium. And we did a task-based experiment as well. This is called the NBAC task. It's where a series of cards is shown to a patient, and they're told to click a button whenever the card that they see is either the same as the previous card or the same as the card shown 2 times ago. It's quite a difficult task to do, especially in the 2-back form. And what we found was that in non-surgical controls, there was not really much change from before to 6 weeks after surgery in connectivity within the posterior cingulate or the dorsal posterior cingulate. However, in surgical patients, there was an increase in activity and connectivity in this brain region, specifically while the patients were doing this task, the NBAC task. Now, this is bizarre because I told you that the default mode network is task negative. It's supposed to be off when somebody's doing a cognitive task. Yet here in the surgical patients, after surgery, their DMN, and particularly the dorsal posterior cingulate, is more active when they're doing a task, exactly when it should be less active. And if you graph this and compare it to global cognitive change, you see a similar relationship as I showed you before in the resting state data. The more of a drop in cognition that the patient has here on the x-axis, the greater the increase in task-based functional connectivity within the dorsal posterior cingulate, exactly the opposite of what should be happening. That's in the surgical patients. In the non-surgical controls in blue, there's really just a cloud and there's no significant relationship between these variables. Regardless of whether they improved or declined in cognition, there's no relationship with changes in connectivity within the dorsal posterior cingulate. So to just conclude here, I think we've covered the fact that DMN activity and connectivity is altered in patients with delirium and in those with cognitive dysfunction following surgery and anesthesia. The DMN changes in these disorders are complex and they depend on the patient's cognitive focus. We see lower posterior cingulate activity at rest when it should normally be active in patients after surgery, and vice versa. We see this brain region becoming more active in these patients when it should be less active when they're doing a task. And finally, an important future direction for this work, both in the delirium and cognitive dysfunction space, is the question of what really causes these postoperative changes within DMN connectivity and activity. One hypothesis is that these might be caused by neuroinflammation, and that's something our group is looking at now in our Intuit study. So with that, I'd like to just give thanks for our funders and thank you all for your attention. Thanks, George. Much appreciated. Thank you all for coming.
It's good to see you all.
Yeah, even virtually.
Yeah, great job, everyone. Those were excellent presentations, showing us the rhythms of life from circadian to alpha Berger waves. Good job.
Thank you.
Thank you. And for those who didn't quite catch it, Dr. McIver has a little bit of a joke in there in calling alpha waves Berger waves, since they were named so, not by me at all, but by Hans Berger, who is a German, I guess, doctor and scientist who discovered these a long time ago. No relation as far as I know. We'd be happy to take any questions as folks are coming in for any of the speakers. One question I had for Kendall, actually, that she already sort of answered in the chat before was, I didn't quite catch what kind of samples it was. It was whole blood that was used in her work. And there's a question in the chat for Phil. I'll let Phil go ahead and take that.
Sure. So the question is, do we see any burst suppression associated with delirium? And we, in the studies I presented, we didn't specifically test burst suppression or EEG suppression with delirium. But yes, I mean, this is something many groups have studied. It does seem like that there is an association between time in suppressed states intraoperatively and risk of postoperative delirium, particularly when patients enter into suppressed states at lower anesthetic depths. And so one hypothesis that's out there is that patients who are more likely to enter suppressed states are more at risk for postoperative delirium. And we have in separate studies seen suppression and slow wave connectivity states associated with delirium. So yes, that association is certainly there.
Great, thanks, Phil. Other questions?
I think I see Ben's hand up. It's sort of blending in with the background, but.
Oh, was it? Okay, yeah, go ahead, Ben.
Well, thanks for organizing a great panel, and all three speakers had great talks. I have a question for Phil. It's great to see you, Phil. I was wondering about this possibility of caffeine as a intervention for patients who are either developing delirium or prevention against delirium. Do you think many of our cases of hypoactive delirium could be attributed to caffeine withdrawal or deficiencies in adenosine? And do you think caffeine could be a potential rescue therapy for patients in that manner?
Thanks, Ben. Those are great questions. The short answer is yes. I think the connection is certainly there. I mean, not just with the adenosine antagonism, but caffeine, as you know, really increases cortical cholinergic tone, really helps to increase cholinergic output and tone to the prefrontal cortex. And we know that acetylcholine is so important for arousal and cognition, and caffeine is great with boosting cortical cholinergic tone. So in that context, I think mechanistically it's plausible that caffeine can help both prevent and ameliorate and treat particularly the hypoactive phenotype for delirium. And it's certainly worth testing, but I think there's a plausible relationship there, certainly.
Great, thanks, Phil. And it looks like you're popular. You've got one more question in the chat here from Dr. G again.
Great question. No, this is a pragmatic setting. So we did not, in the sense that for really optimizing external study validity, we didn't prescribe the type of anesthetic in the OR. We sort of let anesthetic teams use whatever volatile agent or propofol, as they wanted. So no, we didn't prescribe a certain anesthetic technique, other than that it needed to be general to be eligible for the trial.
Okay, I'm going to have to ask the question instead of typing. Nice job. Okay, so you use a very low dose of caffeine. So 200 milligram of caffeine citrate is like a cup of Starbucks coffee. It's very small. And clearly, depend on what kind of anesthetic you use. If you use a propofol basis anesthetic, it probably the effect for your caffeine at that dose is probably very minimal. But if you use the inhalation agent, because they wash out much quicker, and that dose may be a little more effect. And that's why I asked to say, because yesterday George said that he didn't see any change in the recovery time, like an emergent time, which is kind of understandable because we use a much higher dose for emergence. That's why, and they actually, most of the subject is get up and start doing the psychomotor function test. But we didn't have enough data. Like, I mean, you have a lot more subject than we have, but that's why you may see that change. But most of the subject, if you use propofol, they don't even know they have caffeine. Even they took 500 milligram of caffeine IV. We asked them whether they know they got caffeine or saline. 7 out of 8, they guess it wrong. So that tells you how potent the anesthetic can be if you give them a higher dose. So that's why you may see a little different if you give a little higher dose of caffeine.
Yeah, great points. Thank you very much. And especially in a larger scale setting, I think it's certainly worth testing the interaction statistically with caffeine versus type of anesthetic. Part of the rationale for 200 milligrams for us was also just safety. Just not a lot of data on studying caffeine during emergence, right when there can be hemodynamic perturbations after surgery. And so we kind of opted for a bit of a relatively lower dose just from a safety standpoint. But you're right, from an emergence standpoint, it may very well be that, in the operating room, we might need a higher dose, depending on the type of anesthetic, but also if we're really trying to test emergence time. I think those are very, very valid points.
Thank you.
Interesting. Dr. Smith, it looks like we have a question for you in the chat from Dr. Kells. I'll let you take that.
Yes, thank you, Dr. Kells. Thank you so much for your question. So I think you're getting at, the fact that our area under the curve dropped dramatically on our post-procedural analyses. And I think, there could be a couple of reasons for that. So I think that, I think it's sort of complicated because we think of time signature and we think of accuracy of time signature. There's a couple of ways to interpret that. And what my studies on non-surgical patients showed, that variance that we were seeing in the non-surgical patient population actually sort of, it wasn't really lack of accuracy, but actually it was helping us understand chronotype. So there's sort of a question of whether or not, accuracy is even the right word for that. And maybe time signature is actually giving us an accurate estimate, but that's just very out of sync with true time. But it's sort of an open question. And I think that there are a few other cohorts of patients that we would want to look at to maybe tease that apart a little bit better. Maybe thinking outside of sort of our surgical patient population, maybe patients who are undergoing an anesthetic, for example, for an MRI or TEE or something like that might help us sort of tease that apart a little bit. And I apologize if I didn't answer your question appropriately, but that's sort of what I was thinking.
Great. Thanks, Dr. Smith. Dr. Kells, any follow-up or? See, any follow-up, Dr. Kells or Max? No? Okay. Any other questions right now? If not, it looks like George has some points to add on the caffeine bit.
Thank you. First of all, great session, really exciting work and exciting new directions, so thank you. Just one brief follow-up comment related to caffeine. I think it's important to remember that the difference between the wonderful studies that went on at the University of Chicago, both animal, also in human, and our study is that these were surgical patients. There was polypharmacy, there were comorbidities, there was surgery. And so while it might simply be attributable to dose, and that's something that we can explore and should explore in the future, it could also be due to the fact that this is a different translational phase with a lot of different factors than a healthy volunteer population. Thank you.
Thank you, George. I think there's we don't really know the doses, what is the right dose, particularly for patients. You're absolutely right. So because there's people with like coronary artery disease, you always want to know whether that will change their heart rate and blood pressure. But in our study, we didn't follow the blood pressure and heart rate in volunteer and didn't change at all for a much higher dose. So, I mean, there's a lot of room to improve. That's what I'm trying to say.
I agree, but again, they didn't have surgery, and so that's the
they're healthy. Difference. They're healthy too.
For what it's worth, to point out, when we published this in the appendix, our blood pressure, heart rate were essentially identical in the packet between the groups as well, fortunately, from a safety standpoint. So that was reassuring to see.
Great. Okay, it looks like Ben had a question here in the chat, and he asked about the posterior cingulate and did we carry out any of our analyses on other key DMN, default mode network hubs, such as the medial prefrontal cortex. So, yeah, I'm sorry, I didn't have a chance to discuss the method as much in the presentation. Most of the fMRI work I showed in the last couple of slides was intrinsic connectivity contrast, where you basically ask what voxels across the entire brain have connectivity that's altered with in correlation with some other variable, like cognitive performance or difference between groups. And so it's really looking across the entire brain. And the thing that came out was the posterior cingulate. In the Choi et al. American Journal of Psychiatry paper from 2012, they put a seed in the posterior cingulate, and then what they picked up was that connectivity differences to the precuneus were related to those outcomes. That being said, I think you could probably see in the PET study from Gideon Kaplan's group, there's probably more of a change in metabolism and cerebral or local cerebral metabolic rate within the anterior portions of the DMN than the posterior ones. At least that's what I see in their PET scans. Yeah, I don't know about, you know, what might be a better therapeutic target. Were you thinking about sort of in terms of TMS or direct current or?
I hadn't anything specified, but I think your studies in terms of, you know, where in the default mode network, if there's an area that's really important for the pathogenesis of delirium, that might be a great target for others who are in the position to use different therapies. Thanks a lot, Miles.
Yeah, absolutely. One other quick comment on that is just one thing Jeff Browndyke and I did a lot of the work for those studies have commented on is that the posterior cingulate is actually one of the most metabolically active regions in the brain. So one potential explanation is this may just be an artifact of, you know, it's easiest to see a drop where you already started the highest. And with that, I believe Dr. Mashore has a follow-up comment.
Yes, thank you. Just in terms of the DMN and some of the localization that you and Ben were just talking about, some of the work that we've been doing over the past few years, led by Zerui Huang and Tony Hudetz, show that it's also the temporal circuit, the temporal sequencing, and these transitions across functional brain networks, the correlation or anti-correlation with DMN and dorsal attention networks. And actually, in terms of another site that we found important, and this came out in a Cell Reports publication last year, is actually the anterior insula that seemed to be important in regulating those network transitions and to maintain the integrity of that temporal circuit, if you will. So anyway, I just wanted to throw that out there, and I think that, you know, not only thinking about the spatial circuits, but the temporal circuits and the cycling and the accessibility of these different states might also be helpful as you consider the relationship of networks and delirium.
Yeah, absolutely. Absolutely. Great point. Thanks for mentioning that. Actually, that brings me to another point. Something you said brought up that reminded me of something in Phil's talk. We've never studied caffeine here in a scientific way. The only time I've ever given it in the OR is during awake trainings when the patients just won't wake up and won't follow commands. Interestingly enough, we've had a couple insular tumors, insular resection cases, where we've given a lot of caffeine, at least what I thought was a lot, 300, 400 milligrams. It made the patients sweat. I think their blood pressure went up a little bit, but nothing else happened. They didn't wake up anymore. And so it just makes me think a little bit about the role of the insula, possibly in consciousness, possibly in delirium, and possibly, you know, is there a localized effect of caffeine somewhere within the brain? Phil or George, I'd be interested to hear your thoughts.
It's really interesting. I think that the challenge and confound though as well is right with these intracranial cases where there's a tumor where functional networks can be disrupted, both on the micro scale and macro scale. It's hard to say, right? Anecdotally, I mean, I still remember one patient who we later found out was randomized to caffeine. I mean, he woke up at 0.4 mac of ISO and was awake and with it, and this is 200 milligrams. And I think too, it's worth drawing a distinction between those who are habitual users of caffeine and those aren't, because it really seems like that does make an outcome in terms of early postoperative outcomes. Same with headache, that's actually a pretty major issue. So there's a lot we don't know. There's a lot we don't know, and we're designing some follow-up trials, and so hopefully we'll know more in the coming years, but it's a great question.
Looks like there's another question in the chat from Dr. Rao about using modafinil, the narcolepsy Provigil, I think is the commercial name, for those who have delayed emergence. Phil, do you want to speak to that?
You know, from what I know, I think there have been some single center trials over the last 15 or so years showing some variable benefits. I think the fundamental challenge, and George has spoken to this and others, you know, these aren't just redundant circuits and networks, right? I mean, there are very specific, it seems that there are very specific functions to each of these circuits and neurotransmitter systems where, you know, different groups have stimulated different nuclei with different neurotransmitter systems and have had different results with respect to emergence patterns in both basic science models and clinical models. And so that might be reflected with the variable outcomes we've seen with modafinil. So I think the bigger question is just trying to sort of better understand these circuits and neurotransmitter systems and sort of how they sort of feed into emergence patterns and wakefulness.
Yeah, absolutely. Let's see here. Comment from Ben. Caffeine antagonizes both A1 and A2A adenosine receptors, which have differential expression patterns within the brain. Absolutely. One question actually for you guys who've worked on caffeine. I remember from longer ago than I care to admit, when studying for the MCAT, that caffeine at least used to be thought of as a phosphodiesterase inhibitor in addition to an adenosine antagonist. Is that still considered to be part of its mechanism of action? Or is that not really viewed as so prominent anymore?
I don't know. It's a good question.
Can you say that again?
I think theophylline is, right?
Yeah, theophylline is.
Mm-hmm.
Well, can you say the question?
Yeah, the question was whether caffeine really works as a phosphodiesterase inhibitor in addition to its role as an adenosine antagonist.
No, that's exactly right. That's why they said it's the two mechanisms. One is to increase because they slow down the breakdown of cyclic AMP. And that's what we found and we presented yesterday. It have a significant effect. So the other, I mean, obviously there's other effect on the adenosine receptor, increased calcium, intracellular calcium, and all kind of other effects. But I mean, it's not a pure drug. It can be dirty if you give a lot.
Right, right.
Yeah.
As a follow-up to that, since you mentioned it, is it known whether the cardiac and vascular effects are due to the adenosine mechanism versus the phosphodiesterase mechanism?
I mean, if you give a phosphorine and you give phosphorine, basically is stimulate the increase the cyclic AMP production. In that case, actually you can have increase your contractility if you have increased cyclic AMP. That's how they treatment the congestive heart failure.
Okay, great. Other questions for any of the speakers? If not, I have one more question for Kendall. And this is not my field, but I recall from years and years ago reading a paper in anesthesiology that showed something about the length of time somebody was under a GA did not relieve any of their sleep debt. So if somebody stayed up all night and then was anesthetized, it didn't relieve the sleep debt at all. And the other thing is subjectively, patients, and I've experienced this myself, when you wake up from anesthesia, you don't have the sense that you are asleep for some period of time like we do with normal sleep. You wake up and it's as if the entire world ceased to exist for a period and then it just reappeared. Do you think that that means that it causes a phase shift, or what do you think that means about what anesthesia does to circadian phase?
Yeah, I think that's a really good question, Miles. And the short answer is, it's still an open question. There is a group at the University of Auckland led by Dr. Guy Warman. And he's really interested in this question, and his studies in honeybees and mice actually show that there is a significant phase delay after prolonged general anesthetics, specifically isoflurane. And so And he's got a really interesting clinical trial actually taking advantage of kidney donor patient, healthy kidney donor patients. And they're actually also demonstrating a phase delay. And he's looking at whether or not blue light can actually rescue that. And so I think that's a really, really interesting question. I'm really excited about sort of looking at that further, and then, you know, what relationships there might be with neurocognitive disorders perioperatively. And sort of that does sort of intuitively go along with the idea that when you go under anesthesia, the idea that there was no passage of time, you know, and that maybe there is sort of some type of disruption in our circadian clocks that would result in a phase delay. And so that's sort of why our hypothesis is that, you know, we would see a phase delay, but sort of, you know, we're still in the data collection phase, and we'll kind of see what it looks like. But I think certainly, I mean, it's not surprising to think that circadian rhythms will be disrupted after major surgery. Yeah, it's super fascinating. Yeah.
Absolutely. I see in the chat that we're going to get kicked off in 5 minutes, which I was wondering if that was just going to happen spontaneously at some point. So it's good to know when it'll happen. Interesting point from Max Kells in the chat that increasing CO2 or hypercarbia can accelerate emergence, in part through enhancing orexin signaling. So I can't help but follow up, Max, with a question to you. Do you think there's any role for orexin disturbances in post-op delirium, in especially the circadian or the sleep-wake disturbances we see in patients with delirium?
It's possible. To my knowledge, that hasn't really been studied well yet. There's certainly a cohort of narcoleptics that have delayed emergence, but in my limited experience with them, when they do emerge, I haven't seen delirium present in them.
Gotcha. And George Mashore has one more comment, and I can't think of a better person to close out this session than a comment from George. So thank you.
Sorry to be speaking so much, but I'm so engaged and enthralled by these topics. Just you know, one comment on the you know, process C and process S and timing and sleep recovery during anesthesia, at least the basic science studies I just wanted to throw out there. And Max has done work on this, and we have done work on this. You know, there appears to be a dissociation between how non-REM and REM homeostasis, you know, are satisfied during volatile anesthesia. And I know what we found when we sleep-deprived animals and then subjected them to a volatile anesthetic, sevoflurane, versus ad lib recovery. Actually, there was profound slow wave sleep homeostasis that happened during the anesthetic, but REM sleep debt was just held in suspension until the termination of the anesthetic, and then it started to discharge. And so I think that's just another interesting complexity that, you know, there could be different phases of sleep that have distinct patterns, both with respect to homeostasis and circadian rhythm. Thanks.
Great point. Interesting. Thanks for sharing that, George. I was going to say that George would be the perfect person to close this out, but I see one more question in the chat from Valerie Villanueva. I think this question is probably targeted, well, I'm not, probably towards Kendall Smith. Kendall, do you want to take a stab at this?
Yeah, I think it's a great question. And I think this is where, I mean, I think it sounds like you're asking about how to sort of separate the response to surgery versus the response to anesthetic exposure. I think this is where, you know, having a, you know, a control per se that doesn't have surgery, but that it has anesthetic exposure might be helpful in helping to tease that apart, because I agree. I mean, a longer surgery would mean increased inflammation, and that would be a little bit more difficult to tease out. But feel free, anybody else who wants to chime in on that one.
Yeah, I think that's a great point. Well, thank you all for coming. It looks like we're going to get cut off any moment here, but it's really great to see you all. A message from the heart as well as from the brain. Take care, everybody.
Thanks, everyone.
Bye. Thank you.